Hey everyone, Dr. D here, and in this video we are going to cover Chapter 14, the genetic material. Remember that DNA is a nucleic acid. This should be a review that DNA is a nucleic acid made of nucleotides. Nucleotides consist of a 5-carbon sugar called deoxyribose, also a phosphate group and a nitrogenous base. Remember, DNA has A's, G's, C's, and T nitrogenous bases. A's, G's, C's, and T's. And you also have a 5' end. Let me show you. A 5' end with a phosphate group, a 3' end with a hydroxyl group. So don't forget DNA. This should be a review. DNA has a 5' end with a phosphate group and a 3' end with a hydroxyl group, a deoxyribose, and a nitrogenous base. Remember, you got the purines, adenine and guanine, and the pyrimidines, cytosine, thymine, and uracil. And do you remember how nucleotides link together? Dehydration reaction occurs between the 5' phosphate group of one nucleotide, this nucleotide, the 5' phosphate group, and the 3' OH of this nucleotide. And those bonds are called phosphodiester bonds. Now, we can thank this lady, Rosalind Franklin, for our understanding of double-stranded DNA structure. She used a technique called X-ray crystallography to figure out that DNA is a double helix. And her boss, Maurice Wilkins, were pioneers in that field. Now, what you should realize is a lot of the evidence was used by James Watson and Francis Crick, but these researchers did not give credit to Rosalind Franklin, who actually did the experiments to figure out the structure of DNA. So although they won the Nobel Prize, they didn't actually perform a single experiment themselves. They actually kind of took Rosalind's work. So now we know that DNA is a double helix. Remember, you have the phosphate sugar, phosphate backbone. DNA always has a 5' end, where there's a phosphate group on the single-strand DNA, and then a 3' end with a hydroxyl group. Remember that? DNA is directional. You got the sugar phosphate backbone, then you got the nitrogenous bases, the purines and the pyrimidines, pointing to the side. And this forms a double helix, right? Two strands, or I should say strands. Two strands come together and form a double helix. And those two strands are what is known as complementary. What does that mean? T's pair with the complement A with 2 hydrogen bonds. G's pair with the complement C with 3 hydrogen bonds. Okay, and don't forget, this is very important for this chapter, that DNA, the 2 strands are anti-parallel. So look at this strand of DNA here on the left. Its 5' end is up here. But if you look at the complementary strand on the right, the 5' end is down here. Why? Because the 2 strands go in opposite directions. They are anti-parallel. Don't forget that as well. Okay, again, G's pair with C's through 3 hydrogen bonds. A's pair with T's with 2. Now, this chapter really focuses on how DNA gets replicated. Okay, it has to do with how DNA gets replicated. And all you need to know about in this slide is that there were different proposed ways that DNA gets replicated, and then researchers did studies to figure out that actually it's the middle way. You see this middle way here? It's a semi-conservative way. This is the way that you should know that DNA gets actually replicated. What does semi-conservative replication of DNA mean? It means that when you have a template DNA, double-strand DNA, those 2 strands separate from one another. Okay, that's the blue strand here and the blue strand here. And each blue strand serves as a template for synthesis of a new strand. So the peach color or orange color strand is the new daughter strand. Okay, so we're going to talk a lot about how DNA gets copied. Every time DNA gets copied, remember you've got a template strand or parent strand, and then you've got a daughter strand. This is the strand that's newly made or being synthesized. See, so in green you have the newly synthesized strand, and in blue you have the template strand. Now look, this is how DNA is made. This is how DNA is copied. You are copying the blue strand and making the complement, which is the green strand. And the enzyme responsible for this copying is called DNA polymerase 3. In prokaryotes, it's called DNA polymerase 3. This enzyme carries along and adds the correct complementary nucleotide to the strand. So for example, you see here on the template, what do we have? Adenine, we have A. What complements A's? It's T's. Okay, so what does DNA polymerase 3 do? It grabs this thymine and it attaches it here to the 3' end of the daughter strand of DNA. You see the 3' OH right here? Well, DNA polymerase 3 attaches thymine to that 3' OH. Okay? Now, you might be wondering, how come this thymine has 3 phosphate groups? I don't really need you to know that, but if you want to know, these 3 phosphate groups are required for attachment of this nucleotide triphosphate to this 3' OH. 2 of those phosphates get hydrolyzed off, and the energy released is used to form that phosphodiester bond. Okay, so what would DNA polymerase 3 go on to do next? It would again move one more nucleotide over. It would find another adenine, complement that with another thymine triphosphate, and continue linking the complementary nucleotides until it's done copying the template into the complementary DNA. Okay, so one very important thing you need to understand in this chapter. Do you notice how the DNA is being synthesized? The new strand is being made in what direction? From the 5' towards the 3'. Okay, looks like Tig's on his way out. Okay. Hello, everyone. All right, so what you need to know is what it says at the bottom here. DNA is synthesized in a 5' to 3' direction. What does that mean? Take a look. The daughter strand is being synthesized from 5' towards 3'. DNA polymerase 3 can only add nucleotides to the 3' end of the growing strand of DNA. It cannot add nucleotides to the 5' end. All right, so how does DNA actually get copied? What you need to know is that DNA polymerase 3, which is the enzyme I've been talking about over here, it's great for copying DNA in a 5' to 3' direction. However, it has one limitation, and that is that if you have a strand of DNA, you can't start the second strand with DNA polymerase 3. So DNA polymerase 3 is not capable of starting a new strand. It needs something to build off of. So in that case, this enzyme called RNA polymerase or primase, it must attach first, put down a short stretch of complementary RNA, detach, and only then can DNA polymerase 3 attach and copy the rest of the template into complementary DNA. So do you notice what happened? The red oval here is RNA polymerase. It attaches to the single strand DNA. It starts a short strand of RNA. The yellow is RNA called the primer. Okay, this short stretch of yellow is called the primer. You have to put down a short stretch of RNA primer first. Then the red oval, the RNA polymerase, or aka primase, detaches, allowing the blue oval, DNA polymerase 3, to attach. And now DNA polymerase 3 would have something to build off of, this primer, and it would build the rest of the DNA into DNA. It would complement the DNA into DNA. So let me show you a quick little video of me at the board explaining this process, and we'll be right back.
All right, so here I'm going to explain how we copy DNA. So if we have a single strand of DNA, what you need to understand is this. Something needs to happen. You would think DNA polymerase 3 would attach to the DNA and copy the DNA, right? But that's actually not what happens. A different enzyme called, I'm going to draw it here, a different enzyme called RNA polymerase, also known as primase. This enzyme attaches first and actually copies in a 5' to 3' direction. It copies the DNA into a short stretch of RNA. Why is this? Because DNA polymerase 3 is not able to start a new strand from scratch. DNA polymerase 3 needs something to build off of. So what does RNA polymerase do? RNA polymerase attaches to the DNA and copies the DNA into RNA, a short stretch of RNA, and then the RNA polymerase goes away. All right, the RNA polymerase goes away. Next, what happens is DNA polymerase can now attach and take over, because now DNA polymerase has something to build off of. You see, DNA polymerase has something to build off of. And remember, you are building 5' to 3'. You're always building the DNA 5' to 3'. Okay, so just remember, when you're dealing with copying DNA, first RNA polymerase has to put down a short stretch of RNA, and this is called the primer. This short stretch of RNA is called the primer. Next, DNA polymerase 3 can take over and copy the rest of the DNA into DNA, right? Complementary DNA.
All right, so now we know how DNA gets copied. And, oh, guess who's back? You would never guess. It's Tig. Welcome back, Tig. He's stretching. He's having a good time. All right, we're about to explain some DNA replication. All right. Welcome back, Tig. You were missed. You were missed. All right. So, what's going on? Tig needs pets. And back. So, now we know how DNA gets copied. First, you have to put down an RNA primer, which is placed by RNA polymerase, also known as primase. Okay. Then DNA polymerase 3 is able to attach to the DNA and copy the rest of the DNA into DNA. Okay. So let me skip this slide for a second. We'll come back to this. All right. So what you need to understand is DNA is double-stranded, right? It's not just one strand of DNA. So the first thing you have to do is unzip the DNA. All right, you got to unzip the DNA. So you've got 2 strands of DNA to copy. And this here is called the replication fork, by the way. You see, it's like a fork in the road. Okay, so once you have unzipped the DNA, then you get to copy the DNA. You have to copy the DNA. And this happens in 2 different ways. It happens in one direction on one strand, and in the other direction on the other strand. So for this explanation on how DNA, the 2 strands of DNA get copied, I'll have to take you back to the board. Let's go back to the board.
All right, so how is DNA copied, double-stranded DNA? Let's take a look. So here we have double-stranded DNA, right? You've got your double-stranded DNA. These are the nucleotide pairings. And you want to copy the DNA. Remember, double-stranded DNA is copied in a semi-conservative fashion. So what we need to do is separate the 2 strands of DNA from one another. Okay, so imagine an enzyme is going to come through, cut those hydrogen bonds, separating the 2 strands from one another. Okay, what you're going to get is something like this. Okay, that's the 3' end of this strand, and this strand, that's the 5' end of the other strand. Remember, DNA is anti-parallel. That's why this end is the 3' end of this strand. That's the 5' end of the other strand. Okay, so we need to copy this DNA, right? So let's talk about how we're going to copy the bottom strand first. Remember, you have to synthesize DNA in a 5' to 3' direction, right? With respect to the new strand of DNA. So we need to copy which way? Are we going to copy this way, or are we going to copy this way? 5' to 3'. Actually, it would be this way, right? We'd need to go in this direction for 5' to 3', because the new daughter strand is going to be anti-parallel to this bottom strand. So, can DNA polymerase attach and start copying the DNA into complementary DNA? No. Remember what we just talked about before this? RNA polymerase. Remember, RNA polymerase must attach first. It then copies a short stretch of the DNA into RNA and then leaves, right? The RNA polymerase goes away, leaving behind a short stretch of RNA. Red is RNA. And this short stretch of RNA was built 5' to 3', and that short stretch is called the primer. The primer. Okay? But now that the RNA polymerase is gone and it's left behind the little short primer, now DNA polymerase is able to attach. Okay, let me show you that. This marker is fighting me. DNA polymerase now attaches and it copies the rest of it into DNA, right? And it just copies towards the replication fork. Okay, now this is called, this bottom strand is called the leading strand, and I'll explain why in a second. It's the leading strand. What does that mean? The leading strand is the strand that is being synthesized toward the replication fork. You notice how we're going 5' to 3' towards the replication fork. The more you unzip the DNA, the more you simply copy the DNA towards the fork. So on the leading strand, you're building towards the fork in a 5' to 3' direction, and also you only need one primer. That primer's at the very beginning. Okay, so let me explain the other strand. So if I want to copy this other strand, what do I need to do? Can RNA polymerase attach here and copy towards the fork? The answer is no. If you paid attention, you know that you can only make DNA 5' to 3'. So that would be this direction, away from the fork. Does that make sense? So RNA polymerase still needs to attach, but it attaches right near the fork and it builds the RNA away from the fork. RNA polymerase then detaches and DNA polymerase can attach, and DNA polymerase now has the primer to build off of, and it will build the rest of the DNA away from the fork. Okay, that's called the lagging strand. This one's called the lagging strand. Why? Because you are lagging behind. You are moving away from the replication fork. Why? Because 5' to 3' in this case is away from the fork. All right, so what happens when you continue? Let's say I unzip even more DNA. Let's say I unzip even more of the DNA. I'm just showing you what happens when I continue to unzip the DNA. What's happening on the leading strand? That DNA polymerase doesn't care. It just keeps continuing to copy towards the fork. What happens on the lagging strand? RNA polymerase must attach right here by the fork, build a short stretch of RNA called the primer away from the fork. DNA polymerase 3 then attaches and copies the rest into DNA. And by the way, DNA polymerase 3 will stall out here when it touches the previous primer on the previous fragment. And by the way, on the lagging strand, so every time you unzip the DNA, you got to put down a new primer and more DNA. You unzip the DNA, put down a new primer and more DNA. Okay, and you're going to leave behind these fragments of DNA and RNA. This is a fragment, and these fragments are called Okazaki fragments. Okay, all right, so in the leading strand, you put down one primer, and then you build DNA towards the fork. The more you unzip the fork, the more you copy DNA. On the lagging strand, you unzip first, then put down a primer away from the fork, and then DNA away from the fork. When you unzip more, you have to put down a whole new primer with more DNA. So you're going to end up with a bunch of these fragments of RNA DNA on the lagging strand, and those fragments are called Okazaki fragments. So I hope that helps. That's how the leading strand and the lagging strand are copied during DNA replication.
All right, so at this point, we understand how DNA replication works on double-stranded DNA. Remember, you unzip the two strands, and one strand is being synthesized toward the fork in a 5' to 3' direction. That's called the leading strand. And then the other strand is being synthesized away from the fork, and that's called the lagging strand. And remember, these fragments that result are called Okazaki fragments. All right, so there's a few things we need to touch on as well. I said there's an enzyme that unwinds the DNA, basically unzips the DNA, if you could think about it that way, so you end up with two strands. Let me tell you what that's called. That's called helicase. Helicases are enzymes that use energy from ATP to unwind the DNA. So the helicase enzymes are the ones unzipping that DNA. And once unzipped, let me show you, once those two strands are apart, what they want to do is reanneal. They want to come back together and re-zip, okay? So to prevent that from happening, a bunch of these single strand binding proteins, SSBs, bind to the strands of DNA and keep them apart. Okay, that's what they do. Okay, what else? Now, one more thing to talk about. Once you have on the lagging strand, let's take a close look at the lagging strand here. Here's the lagging strand. Do you remember what happens on the lagging strand? You have these primers that form from the fork, away from the fork by primase, remember, also known as RNA polymerase. The primase then detaches, allowing DNA polymerase 3 to attach and build DNA, which would be colored blue here, build DNA until the DNA polymerase 3 bumps into the previous Okazaki fragment right here. So that results in a bunch of these fragments, doesn't it? Of these Okazaki fragments. Well, what you need to know is after all this happens, you want to get rid of this RNA primer. See this yellow RNA primer? You don't want it there later on. So this other enzyme called DNA polymerase 1, its job is to come in after the fact and remove these primers, remove that RNA, by excising the RNA nucleotides. And you know what it does? Once it removes those RNA nucleotides, it replaces the RNA with DNA. It puts down DNA. Now, when it does this, it does a good job. However, it leaves a nick. It leaves, it's missing a phosphodiester bond in the backbone of the DNA strand. What does that mean? It means it did a good job chopping out the primer and replacing it with DNA. However, it didn't link the two Okazaki fragments together. That's the job of this other enzyme called DNA ligase, which attaches to the DNA right where the nick is and not replaces, but creates a phosphodiester bond linking the two Okazaki fragments once and for all. So this is called lagging strand synthesis. On the lagging strand, primase or RNA polymerase synthesizes a primer in a 5' to 3' direction away from the fork. It then detaches, allowing DNA polymerase 3 to attach and have something to build off of. It will build DNA, which is in blue. It will build DNA until it runs out of space, until it either runs out of DNA to copy or until it bumps into a previous Okazaki fragment, and then the DNA polymerase detaches. And remember, that DNA polymerase is called DNA polymerase 3. DNA polymerase 3. It detaches. Then DNA polymerase 1 attaches. It chops out the primers and replaces the primers with DNA. And finally, to link the Okazaki fragments, DNA ligase attaches, linking the two Okazaki fragments. Okay, so that is the process of lagging strand synthesis. What else do we need to know about? How about this guy, DNA gyrase? You see this guy, DNA gyrase. Notice it's an enzyme that's working up here, well ahead of the helicase, well ahead of the fork. You see this is that replication fork. Gyrase is doing its job way up here. Let me explain how gyrase works. Gyrase, you see DNA gyrase or topoisomerase, it works by, the definition is it corrects overwinding ahead of the replication forks by breaking, swiveling, and rejoining the DNA strands. So what does that mean? Let me explain it to you this way. Imagine if I have a zipper, right? If you have a zipper, you could unzip it, you could rezip it, you could unzip it and rezip it, right? It's a zipper. You know, it's easy to unzip, rezip. But imagine if I took my zipper and I attached it to my table, right? So I take part of the bottom part of my zipper and I glue it to my table. Okay, so now I have a zipper that's glued to a table. Now, what if I took the rest of the zipper and I twisted it and twisted it and twisted it? So now I have this twisted up zipper, right? It looks like a double helix, kind of like DNA is a double helix, right? Now, what do you think would happen if I tried to unzip my twisted zipper? Would it unzip? It might unzip a little bit, but then it'll stop because the rest of it's twisting up so much. It's called supercoiling. The rest of it's so supercoiled, I can't unzip anymore. Does that make sense? This is exactly what happens in DNA as well. DNA is kind of like a double helix. Well, it is a double helix, but it's kind of like a zipper that's been twisted around too much. And helicase enzyme is trying to unzip that, right? So what topoisomerase does, or gyrase, what that does, imagine if I have a buddy over here with scissors, and my buddy comes in with scissors, cuts the zipper, allows the zipper to uncoils. Allows it to untwist. Then my buddy glues the zipper back together. Now, do you think I could unzip a little bit further? Yes. If my friend's here with scissors cutting the zipper, letting it untwist, gluing it back together, if he keeps doing that, I can continue unzipping forever, right? I can continue unzipping the DNA, and that's exactly what topoisomerase does. Good. What else do we need to talk about? Well, here's another one. Look at this. This is actually more closely. This is more closely showing you how DNA replication occurs. Remember, you've got in green, you have the double-stranded DNA that you're trying to copy. The enzyme here, helicase, unwinds the two strands, unzips the DNA. And then you have those single-strand binding proteins. You see each one of these blue dots is those single-strand binding proteins keeping the two strands apart. And it looks like the top strand here, see the arrow? You're building DNA towards the fork, so this top strand is the leading strand. And then notice that on the bottom strand, the arrow is going away from the fork. So it looks like you're building away from the fork, and that's called the lagging strand, All right, and it looks here, look over here. This appears to be primase or RNA polymerase putting down a primer, going away from the fork. And then once it does that, this blue oval here, called DNA polymerase 3, can build the DNA away from that, okay, starting at that primer, and it synthesizes away from the replication fork until it bumps into the previous primer, okay, forming those Okazaki fragments. And then do you remember what happens next? DNA polymerase 1 excises those RNA nucleotides and replaces them with DNA. And remember, it does a good job, but it doesn't quite link the two fragments together. So DNA ligase attaches to make that final phosphodiester bond, linking the two fragments to one another. Okay, so we just talked about gyrase as well. What gyrase is doing? Remember, it's cutting, unswiveling, and then rejoining the double-strand DNA ahead of the replication fork, like my buddy with scissors and glue, right? Allowing the helicase to continue unwinding the DNA. So we understand 80 or more % of this image based on what we've talked about. What we haven't talked about is this thing that kind of looks like an octopus in this image. This is called the clamp loader or beta clamp loader. And what the clamp loader does is it loads these things. These are proteins called open beta clamps. Okay, so you've got beta clamps, and here's the clamp loader. These are the clamps that it loads. Okay, and look, the clamp loader can load clamps here, you see, next to this DNA polymerase 3, or it can load clamps here. You see, here's a clamp next to this DNA polymerase 3. So all you need to know about the clamp loader is that it can load clamps, and it loads clamps next to the DNA polymerase 3. And the reason these clamps, these beta clamps, get attached next to DNA polymerase 3 is because they, all right, increase the processivity of DNA polymerase 3. The beta clamps increase the processivity of DNA polymerase 3. What does that mean? They keep it attached. Look, so long as there's a beta clamp next to DNA polymerase 3, DNA polymerase 3 remains attached to the DNA, and it continues to do its job. You see here's a beta clamp next to this DNA polymerase 3. DNA polymerase 3 can do its job, copying the DNA into complementary DNA strand. All right. Do you think the leading strand needs more than one clamp? Think about it. The answer is no, because once you put this clamp on, the DNA polymerase 3 is firmly secured to the leading strand, and it continues to copy towards the fork. And the more you unzip, more helicase unzips the DNA, the more DNA polymerase 3 copies DNA towards the fork. However, on the lagging strand, look at the lagging strand. The DNA polymerase 3 is being, it's synthesizing DNA backwards. And once it bumps into this other Okazaki fragment, that's when you would want to remove the beta clamp, which would allow DNA polymerase 3 to detach from the DNA. This tentacle from the beta clamp loader would then swivel, attaching the DNA polymerase 3 in front of this primer, where it would get a new clamp, and it would start over again. It would build this next fragment. The DNA polymerase 3 would be building the next fragment. And every time you build a new fragment, a new Okazaki fragment, the beta clamp would get removed, and the DNA polymerase 3 would detach from the DNA, swivel, get a new clamp, and build the new fragment. All right. So there's just one more concept to talk about, and that is the difference between eukaryotic DNA replication and prokaryotic DNA replication. So how does this process work in eukaryotes, and how is it different than in prokaryotes? Well, let's talk about it. First of all, do you remember prokaryotic DNA replication? We kind of touched on it when we talked about binary fission. Do you guys remember binary fission? You've got a prokaryote which has a single circular molecule of DNA, double-stranded DNA, and replication begins at one origin of replication, ORI. Do you remember that? So here's your double-stranded DNA. At this one spot, that's where the DNA replication begins, at the origin of replication, and you would have replication proceeding in this direction and replication proceeding in the other direction at the same time. And if you're wondering what replisome means, let me show you. Replisome simply means all of these proteins. You see this fork with the helicase and the beta clamp loader and DNA polymerase 3 and primase. You see all of these players here? This is called a replisome. So starting at the origin of replication, you've got one replisome with its own helicase headed in this direction. You've got another replisome with another helicase headed in the other direction. And you can see the blue strand is the template strand and the red strand is the daughter strand being synthesized in both directions at once until both of those helicases run into one another at the termination or the terminus site, resulting in two new strands of DNA. Remember, DNA is replicated in a semi-conservative fashion, meaning you've got an old strand in blue in this case and a new strand or daughter strand in red in this case. Okay, So that's how DNA replication occurs during binary fission, when you're copying your chromosomes. When you're copying your chromosomes in a prokaryote. But what about eukaryotes? Don't eukaryotes have linear DNA? They don't have a closed circle, do they? No, eukaryotes have linear double-stranded DNA chromosomes, and they usually have more than one chromosome. So, for example, humans have 46 linear double-stranded DNAs as our genome. Now, what you need to understand about how eukaryotes are different than prokaryotes is this. First of all, we have a lot more DNA to copy. So we need multiple origins of replication. Okay, we need, see what it says here, multiple replicons. Eukaryotes, because we have so much more DNA to copy, it does not make sense to start copying the DNA at one spot and have one origin of replication. In eukaryotes, we have multiple origins of replication. Not just one. That's what you need to understand. We have linear DNA. We have multiple chromosomes. Those chromosomes have a lot more nucleotides to copy. So we have to start at multiple spots at once. We have multiple origins of replication. Okay. Otherwise, the basic enzymology is similar. You know what that means? That means how prokaryotes had DNA polymerase 3 and DNA polymerase 1 and all this, and primase. Eukaryotes have similar enzymology. Now, I'm not going to go into how exactly the differences work between prokaryotes and eukaryotes, so don't worry about that. But there's one other thing to understand about eukaryotes, and that's this concept of telomeres. In eukaryotes, you have telomeres. And telomeres are simply the names for the ends of our chromosomes. So let's say we have a human chromosome. Let's say human chromosome 1. It's a long piece of double-stranded DNA, right? You know how there's this end of it and then this end of it? You've got two ends to the chromosome. Those ends are called telomeres. The ends of our linear chromosomes are called telomeres, all right? So take a look. This is showing you this is showing you double-stranded DNA after DNA replication is finished. Okay, this is double-stranded DNA after replication is finished. The green strand is the template strand. You see, the green strands are the template strands. And then the blue strands are the daughter strands, the newly synthesized strands. So after DNA is finished, you've got two double-strand DNA molecules. Basically, this is what will be called sister chromatid pairs, right? This is a sister chromatid to this one. However, do you remember that there's still primers in there that you need to replace? So DNA polymerase 1 would come along and chop out this primer. It would chop it out in a 5' to 3' direction and replace it with DNA, right? And then DNA ligase would come in and attach the two Okazaki fragments. But here's the issue with our telomeres. Do you remember what telomeres means? That's the ends of our chromosome. So look at this. This would be a telomere, the end of the chromosome. Imagine if DNA polymerase 1 were to come along. What would it do? DNA polymerase 1 would remove this primer, remove all the gold, right, which is RNA primer. And then it would try to replace it with DNA, right? Could it do that? Think about that for a second. If DNA polymerase 1 removed this primer at the telomere, could it replace it with DNA like it did over here? Well, let's look at what you could do over here. DNA polymerase 1 comes in, removes all these nucleotides. It has something to build off of, so it builds the DNA, the complementary DNA, in a 5' to 3' direction. What if it came over here? What if DNA polymerase 1 removed this primer? Well, here's the problem. It would want to build from right to left, right? Because that's 5' to 3', but there would be nothing to build off of. Do you see this? Look, if DNA polymerase 1 removed this primer, you would have this overhang, and DNA polymerase 1 would not be able to fill in this telomere with DNA because it would have to build from right to left. There's nothing to build off of. DNA polymerase 1 can't build a second strand from scratch, and then if you don't deal with that issue, the chromosomes get smaller and smaller. The chromosomes could actually end up getting shortening the next round during the next round of replication, DNA replication. That's a big problem, right? And that's actually linked to aging. Did you know that during aging, one of the issues that happens during aging is that your telomeres shorten during aging. What does that mean? As you age, your levels of telomerase go down, and which I'm going to explain in a minute what telomerase does, and your telomeres end up getting shorter. So what is telomerase? Telomerase is let me show you. Telomerase is the enzyme that makes telomere section of the lagging strand using an internal RNA template. So that's a fancy way of saying telomerase is the enzyme capable of filling in this overhang, filling in this blank with DNA, preventing the shortening of our telomeres. Did you follow that? Telomerase fills in this telomere with DNA nucleotides, preventing shortening of our telomeres. Okay, and why is that important? Because again, if telomerase wasn't around, our telomeres would shorten with each round of cell division, with each replication cycle, and that's linked with aging. In fact, like I said, as you age, your levels of telomerase go down, and your ability to fill in these telomeres with DNA goes down, and your chromosomes shorten with age. Okay, and that's linked with aging. And in fact, conversely, in cancer cells that are growing and growing and splitting all the time, very youthful, immortal cells. These immortal cancer cells, they're usually overexpressing telomerase. So telomerase was really a big deal for a while in the early 2000s because people thought it might be the holy grail for aging, some kind of magic genetic silver bullet that if we tackle this telomerase issue, people won't age. But then, through a number of studies, they figured out that, telomerase is a big component of aging, but it is not the key to aging. So, again, what do you need to know for the exam? You should know that the telomeres are the ends of our eukaryotic chromosomes. They are in danger of becoming shortened because DNA polymerase 1 cannot fill in those ends once it's removed the primer. However, lucky for us, we have this enzyme telomerase, which is able to prevent telomere shortening and prevent that the deleterious means bad, the deleterious effects of aging and deleterious effects of telomere shortening, or at least until later in age. So anyway, that wraps up, unless I missed something. Nope, doesn't look to be that way. That wraps up chapter 14. I hope you learned something. We've got one more chapter in the semester, chapter 15, which I'm going to get to in the next video. All right, let's buckle down, study hard, and finish strong. This is Dr. D, and I'll see you next time.